So, good day everyone. We are Group ANACARDIACEAE and our topic is breeding for disease resistant. So, what is disease resistant? According to Kushwaha 2020, a physiological deviation from a normal functioning of the organism caused by pathogenic organism is a disease that may caused by fungi, bacteria, or viruses. The inherent ability of the organism to resist or withstand the pathogen is called resistance. Disease resistance commonly met within the plant kingdom relative in nature. Total immunity being is too rare. Its heredity transmission from parent to offspring is essentially Mendelian, but often polygenic. There are two types of disease resistance. First is the vertical resistance, and the second is the horizontal resistance. Why there are two kinds of resistance? Because there are two kinds of infection, the primary and secondary. Primary or allo infection means that the parasite originates away from the host, and it has to travel to the host. It's the equivalent of an immigrant arriving in an island. Clearly, the first infection of any host must be an primary infection. Next is secondary, or the auto. Infection means that the parasite originates on the host that it is infecting. It has no need to travel. This is the equivalent of an immigrant progeny populating an island. Clearly, secondary infection can be occur only after there has been a successfully primary infection. You may think of the primary or allo infection as being the equivalent of the cross-pollination, and think that the secondary or auto-infection being the equivalent of self-pollination. Vertical resistance can be controlled by primary or allo infection. Only secondary or the auto-infection can be controlled by the horizontal resistance, but horizontal resistance can also control the primary or the allo infection. This is because the plant have horizontal resistance, but many of plants have no vertical resistance. Vertical resistance is controlled by major genes, one or two. These genes are readily transferred from one genotype to another. Its also presence of genes can be determined by exposing the plant to particular races. Example is the leaf rust resistance in wheat. Next is the horizontal resistance. Horizontal resistance is controlled by many genes, each with minor effect. It can control a broad range of races due to in fact that many loci are involved. Difficult to transfer resistance from one genotype to another. Individual genes harder to identify. The example of horizontal resistance is the Fusarium head blight in wheat. For the conclusion, varieties with race-specific resistance generally show unstable but complete resistance to a specific pathogen under the most environmental condition. But a single or few mutations in the pathogen may produce a new races that may infect the previous resistance variety. Varieties with horizontal resistance show the stable but incomplete resistance against the different pathogen, and they vary in their reaction to the pathogen under different environment conditions, and the pathogen will have to undergo many more mutations to complete breakdown the resistance of the host.
Mechanism of disease resistance. Disease escape. Otis Malloy 2005 stated that disease escape occurs when susceptible plants do not become diseased for some reason. This may be due to some anatomical or physical character, such as the occurrence of leaf hairs, thick cuticle, or modified stomata, or they may be environmental, in which conditions are not conducive to disease development. It is a prevention mechanism that causes the host to escape pathogenic infection. Early or late maturity of the crop may prevent physical contact of pathogen with the host. Mechanical and anatomical barrier, such as thick cuticle, waxy bloom on leaves and stem, stomatal regulation, prevent penetration of spores. Ergot, a fungal disease of inflorescence in cereal caused by Claviceps purpurea, does not affect varieties of wheat and barley in which the flower remain closed until pollination occurs. Erect leaves of barley avoid deposition of spores of Erysiphe graminis citricae in contrast to prostrate leaves. Early maturing varieties of groundnut escape early leaf spot infection, Cercospora acadicula, and early varieties of wheat escape rust and loose moth infection. A change in planting season has also been successfully employed as a measure of securing escape. Example given, the leaf rust of sugarcane. Now, let's proceed to disease endurance. Plant pathology stated that, in some cases, the plant is capable of carrying on most of the abnormal metabolic processes in spite of the disease condition without reducing the yield to a considerable extent. It is known as disease endurance or disease tolerance. The host, after being infected by the pathogen, tolerates infection and suffers less damage. It does not result in any substantial decrease in yield. This is brought about the influence of external factors. Plants fertilized with phosphatic and potash manures are more tolerant to diseases. This is the case in wheat against rust infection. Rice crop fertilized by silicates are resistant to blast pyricularia or RACEL in Japan. Wheat crops fertilized by potash and phosphatic manure are highly tolerant to mildew and rust infection. The fertilizer acts indirectly to arrest vegetative growth and promote early maturity, better straw, and strengthening tissues to protect the plant, which form a bulwark against pathogenic invasion, disease endurance.
True resistance. It is the ability of the host plant to resist or withstand the attack of a pathogen. True resistance is inheritable and much less subject to environmental influence. It is specific in character. The basis of resistance may be morphological, functional, structural, or protoplasmic. Functional nature of the resistance is determined by opening of stomata, time of opening of flowers and time of maturity, rate of cork formation, and cambial activity. Structural characters include the proportion of strengthening tissues, fiber content, nature of middle lamella, corky layers, number and structure of stomata and lenticels, and their sizes. Protoplasmic factors controlling resistance are related to cell content and include acids, tannins, anthocyanins, chemical constituents and their proportions, antibiotic activity, and hypersensitivity present in the plant cells, and in addition biological antagonism of the protoplasm of the host plant and pathogen. Resistant varieties can be the simplest, practical, effective, and economical method of plant disease control. The use of resistant varieties can not only ensure protection against diseases but also save the time, energy, and the money spent on the measures of control. In addition to these advantages, resistant varieties, if evolved, can be the only practical method of control of such diseases as viruses, phytoplasmas, wilt, and rust, etc., in which chemical control is very expensive and impractical. Methods of breeding for disease resistance. First, we have the selection, introduction, hybridization, pedigree method. The method was first described by H. Lowy in 1927. Pedigree, a description of ancestors of an individual, and it generally goes back to some distant ancestors in the past. Thus, a pedigree would be described the parent, grandparents, great-grandparents, and so on. The pedigree is helpful in finding out if two individuals are related by descent their ancestry and therefore are likely to have some genes in common. Backcross method. Backcross breeding enables breeders to transfer a desired trait such as transgene from one variety, donor parent or DP, into the favored genetic background of another, recurrent parent or RP. If the trait of interest is produced by a dominant gene, this process involves 4 rounds of backcrossing with 7 seasons. Bulk method. The bulk method of breeding was first used by Nielsen in 1908. This method is also known as mass method or the population method. Bulk population breeding is a strategy of crop improvement in which the natural selection effect is solicited more directly in the early generations of the procedure by delaying stringent artificial selection until later generations. Marker-assisted selection. Genetic engineering. Genetic engineering is the process of using recombinant DNA, rDNA technology to alter the genetic makeup of an organism. Traditionally, humans have manipulated genomes indirectly by controlling breeding and selecting offspring and decide traits.
Steps in breeding for disease resistance. First, you need to know the source for resistance. Then what method of breeding for disease resistance. Then testing for disease resistance and the selection for resistant line. Source of disease resistance. Know the variety. Then wild relatives. Species may be the source for multiple disease resistance. Screening of wild crop relatives can capture the high effective form of multiple disease resistance. Then the related species, mutation, and unrelated organism. Testing for disease resistance. For identification of resistant plant from the susceptible ones, an artificial epidemics is created. The soil-borne diseases. Sick plots are created for testing resistance to such diseases. Diseases that are caused by these pathogens which survive in the soil and in residues on the soil surface are defined as soil-borne diseases. Reduces plant growth, increased cost of significant economic loss to the growers and potential damage to the environment. The airborne diseases. It is caused by the spraying of suspension spores. Seed-borne diseases. Dry spores are dusted on seeds or seeds may be soaked in a suspension of pathogen spores. Any malfunction of hosts or plants, those results from continuous irritation by a pathogenic agent which are carried by seed and transferred to the plants and lead to the development of symptoms.
Now we're going to talk about the advantages and some of the problems of breeding for disease resistance. First, let's talk about the advantages. Resistant varieties offer the cheapest means of disease control because they do not rely on synthetic inputs that causes more money. Resistant varieties obviate the use of fungicides, thus reduce environmental resistance. Effectiveness of resistant varieties is not affected by environmental conditions, as they were selected among the vigorous varieties. It safeguards against the inadvertent release of such varieties that are most susceptible than earlier varieties, as resistive variety crops were made, planned, and bred carefully. In the case of rusts and musts, it is the only feasible approach for disease control, bred for this specific disease resistance. And for the problems in breeding for disease resistance, there comes the resistance breakdown, a limit to the genetic control of crop disease. This was due to the genetic changes in the virus virulence gene. Some of its examples are the vertifolia effect and the boom and bust cycle. The horizontal resistance being durable but difficulty relates to an accurate and reliable assessment of the level of resistance. And sometimes, there's a negative correlation between the yield and the disease resistance. And for the introgression of multiple resistance and varieties against several diseases, it requires meticulous planning and far greater effort than that required for single resistance.